Hey, ladies and gents, how you doing today? Today we're going to look at chapters 2 through 4. Chapter 2 is about basics of chemistry as it relates to biology. 3 is all about water, and 4 is everybody's favorite non-reactive element, carbon. So let's start with our chemistry review. So chemical bonds. Chemical bonds are interactions between atoms due to the behavior of electrons of those atoms. So let's start by focusing on covalent bonds. The prefix co means together. So with covalent bonds, electrons are shared between neighboring atoms. Now, how they're shared may differ. So again, anything covalent, electrons are being shared. Let's jump down with polar. Okay, now if we think of it geographically, if polar is at the extremes of the Earth or at the poles, polar covalent bonds have electrons being shared between partner atoms, but the electrons spend more time at one pole or one end. Okay, so again, polar covalent bonds, electrons are being passed back and forth between partner atoms, but they spend more time with the more electronegative atom. In the case of biology, our two biggies that we're going to deal with are oxygen and nitrogen. So the presence of really electronegative atoms like oxygen and nitrogen cause the electrons to spend more time by those atoms and less time with partners, typically like hydrogen. When that occurs, the more electronegative partner becomes partially negative, which is indicated here in the diagram by this sigma minus. So the electrons, or the partner that gets more time with the electrons, takes on a partial negative charge. And the partner that has less time with the electrons takes on a partial positive charge. And as a result of that uneven distribution of electrons, you have these partially positive, partially negative regions. And as a result of that, when molecules are around one another, they take on this sort of new behavior of a cohesive force sort of pulling them together. Now let's contrast this with nonpolar covalent bonds. With nonpolar covalent bonds, the electrons don't spend more time at one pole or one end or the other. And as a result of a more equal dispersion of electrons, there's no attractive force, there's limited attractive force, I should say, between neighboring molecules. And as a result of that, the molecules, when they're around each other, tend to spread out more. So you can visualize this when you think about water and oil. Water molecules are polar because electrons spend more time with oxygen and hydrogen. So the water molecules pull on one another and form a nice rounded drop. Oil is a type of lipid. They're nonpolar because the electrons are shared equally, so the molecule gets more spread out. Oil drop is more spread out. Now, ionic bonds, this is a different deal. With ionic bonds, electrons aren't being shared. They're being moved or transferred from one partner atom to another. The partner that strips away or takes possession of those electrons becomes a negatively charged ion. The partner that lost those electrons becomes a positively charged ion. And because of that, the opposite charges attract one another, and they're held by this ionic bond or this attractive force. All very nice and good until you get in the context of life. Life exists in water, right? Anything that's alive requires water. So because of that, ionic substances tend to dissolve in water. So as a person, moves through the world, he or she, when you look at them, is mostly held together by covalent bonds. Now, ionic bonds do have their place. We'll get into that later on in the semester. But ionic bonds tend to dissolve in water, and so that can disrupt things a little bit. Now, let's talk about hydrogen bonds. Hydrogen bonds, a bit of a misnomer, because hydrogen bonds are actually an intermolecular force. It's an attraction between molecules, okay? A water molecule, like in your example here, the water molecule has covalent bonds between the hydrogen and oxygen, but it's an attraction between neighboring molecules. And that happens because water is a result of polar covalent bonds. Again, that oxygen is more electronegative, so it draws the electrons toward itself and takes on a partial negative charge. The hydrogens get less time with the electrons, so they take on a partial positive charge. So when you look at that image, the neighboring water molecules will actually pull on one another. Now, this sometimes upsets AP Chem students. What I would like you to do is consider this as a force in biology to be relatively weak and fleeting. Because, think about water. There are hydrogen bonds between neighboring water molecules, but those break and form all the time. So because these hydrogen bonds break and form all the time, it's cohesive. It pulls the water molecules together, but water is still a fluid. The molecules can flow past one another. Another way to sort of interpret or imagine these hydrogen bonds is seen in your DNA. DNA is a double helix, it's 2 strands. And those 2 strands are held together by hydrogen bonds between their nitrogenous bases. Like an A or adenine is hydrogen bonded to a thymine in a DNA molecule. Now, an individual, set of hydrogen bonds between some nitrogenous bases, those will be pretty easy to separate. But if you can imagine hundreds of millions of hydrogen bonds, the overall molecule becomes stable. So the individual interaction between molecules with hydrogen bonds, it's relatively easily broken. But over the course of a large number of hydrogen bonds, you can create a somewhat stable or cohesive molecule. All right, van der Waals forces. I think chemistry books and chemistry classes are referring to these as London dispersion forces, and biology will probably catch up to that soon. They're something sort of, again, enigmatic and really something that's maybe difficult to conceptualize. It's created by large molecules and sort of the random movement of electrons within those molecules. So if you've got this big molecule that's thousands of atoms long, and you've got electrons being passed back and forth, just randomly, the electrons may be spending more time at one end of the molecule for an instant, and then they're spending more time at another end of the molecule for an instant. And what this does is it creates regions or clouds, if you want to think of it that way, of slightly negative and slightly positive regions of this molecule. And what it does is it creates, again, sort of a weak attractive force that, over the case of a large molecule, helps stabilize it. All right. So that's what will happen with big molecules like proteins. They'll fold up, and van der Waals forces will help stabilize that big molecule. All right, shape has a major influence over function. If you think about it like a lock and a key, you can think about it in terms of a foot and a shoe, whatever. But shape influences what things are able to do. And that goes all the way down to the atomic level. Atoms form particular bond angles with other atoms, and that influences the shape of molecules. And because of this, molecules fold up in very particular ways. Here we look at the example of starch. Starch is a big storage molecule. When plants perform photosynthesis, they make glucose. What are they going to do with all that glucose? One thing they can do is pump it down into other regions of the plant and store it as a big starch molecule that's made of hundreds or thousands of glucose monomers. Now, we have evolved the capacity to break down that starch and extract energy from it. We do that using an enzyme called amylase that's found in our saliva. Amylase has a shape that complements that of starch, so it can cut the starch into little glucose pieces and release that into our bloodstream so we can get the energy out of it. Well, you better believe starch and amylase have very specific shapes, and those shapes are influenced or dictated by the interactions between the atoms that make them up. Okay, so shape influences function all the way down to the molecular level. Last idea from this chapter, it's that chemical reactions occur when bonds are broken and formed. So when you break bonds and form new bonds, you can have new constituent pieces arranged in new ways, and you make new molecules that, well, do new things. In this process, the reactants are converted to products. And in most cases in biology, this happens with the aid of enzymes. Enzymes make these reactions happen faster than they normally would to help sort of make things operate fast enough to allow life. But the issue is when you convert reactants to products, if you allow products to accumulate, you can reach an equilibrium in the reaction. And that's not good because life requires a disequilibrium. You are not at equilibrium with the environment. If you were, you'd be dead, right? So what happens is when products accumulate, you got to get rid of them. So that's why we take 15, 20 breaths a minute. That's why we urinate multiple times a day. It's because you have millions of trillions of chemical reactions in your body all the time, and you have to be able to get those products out of there, either by converting them to something else or getting them out of the body, so you don't have products accumulate to any substantial degree, and you can keep driving these chemical reactions of life. All right, that gets us through chapter 2. We'll see you back for